welcome into Pushing the Limits. Today I have a part one of a three-part series with world-renowned telomere scientist, Dr. Bill Andrews, and it's all about what are telomeres and how to use telomeres to cure aging, which is his mission in life, if you like. So from his perspective, telomeres are the protective caps at the ends of our chromosomes that play a really crucial role in cellular aging and overall health. Think of them like the plastic tips on the end of your shoelaces, which prevent the laces from fraying. And telomeres protect our DNA from damage during cell division. So each time a cell divides, telomeres shorten slightly. And over time, as they become critically short, the cell can no longer divide properly and either enters a state of senescence or dormancy, or it dies. Now, this process is a key driver of aging and age-related diseases. And in Dr. Andrews' research, he emphasizes that telomere shortening is not just a marker of aging, but a root cause contributing to everything from wrinkles to chronic illnesses like heart disease and Alzheimer's. So we're going to be focusing in on that today. And this is part one of a three-part series. You're really lucky to have such an incredible scientist to guest to learn from. He's really, really amazing. Before we head over to the show, I just want to let you know we've launched our company, our biotech company, Avum Labs. We're super excited about this. We've been working on this project for the last 2 years. I wanted to make a formulation that was going to be safe for everybody, including my elderly mum who's got all sorts of problems and is very fragile, that was going to support our immune system. So today we're going to be talking about telomeres, but telomeres and the immune system are crucial for the aging process, and they each affect each other. In fact, the immune system affects all of the hallmarks of aging. It's a foundational piece. So we've been working on a formulation for the last 2 years. There's over 15 years worth of clinical research and ingredients that we've put together into this incredible formulation, and we're really excited to get it out into the world, to you guys, to actually really on a foundational level really help you with your health and your immune system. As we get older, our immune system starts to become faulty. We start to get things like autoimmune. We start to have less surveillance for cancer, and we are less able to respond when we get a virus or some sort of bacterial infection, etc., etc. So the aim of this formulation was to really give you back the function of a younger immune system, and we've put together an incredible formulation. We have a world-class scientific advisory team behind this product, and it's called Rejuvenate. And everybody should be on Rejuvenate. Pretty much, it's safe for everybody to take. It's very well tolerated. It helps with gut health. It helps with cellular health, as well as things like your barriers in your body. You know, if anybody dealing with leaky gut or leaky blood-brain barriers or other barriers in the body, then this is something for you. If you want to increase your natural killer cell activity, which are the ones that go out and get the baddies like cancer and so on, then this is for you. It helps with many aspects of the aging process. So from cellular health to gut health to immune system health. So check out Rejuvenate. You can go to my shop. If you go to lisatarmity.com, hit the shop button and put in Avum in the search bar, which is AEVUM, and you'll come up with Rejuvenate, our product. Or you can go to avumlabs.co.nz, AEVUM labs.co.nz. So go and check it out. We're really, really proud of the work that me and the team have done. We've got a bunch of doctors and scientists behind this product, and it's pretty next level. So make sure you do check it out. Right, over to the show now with Dr. Bill Andrews. Well, hey, everybody, and welcome in. Today, we are starting a series, a course with Dr. Bill Andrews on curing aging. Now, Dr. Bill, by way of introduction, is a world-leading telomere scientist, aging scientist, and I've had the privilege of knowing him for a few years because I happen to be good mates with his lovely wife, Molly, Bill, can you give us a little bit of your background before we get into this wonderful topic that we're going to be discussing today about how to cure aging?
Well, the key thing is aging has been an obsession of mine, at least understanding it and being able to do something about it since I was a little kid. And so, I mean, it's like I knew when I was 10 years old, this is something that I wanted to do. My father actually put the seed inside my head originally, and he and I were partners up until his passing in 2015. We were going to conferences together and everything like that, but I'm still focused on getting the work done. So all through high school and college, I was the one starting anti-aging clubs and having small discussions. Sometimes there'd only be 4 people in the group, and we'd be just sitting talking about things like, why do we age? What is aging? What's the whole point of it? Is it important? What is there any reason not to cure it? Things like that. Those are the kind of things we used to talk about. But I just never bought into all the theories that existed about aging. I always say the 2s and 2s don't all add up.
Yeah.
So they're explaining everything. So I came up with my own theories, which I'll be delving into during this series. But I back when I got my PhD, and most of my PhD was based on trying to learn everything I needed to know so that I could cure aging. But when I got my PhD and started looking for what are called postdoc positions in universities that were working, university labs working on aging, I found out that everybody, I got accepted everywhere I went. I applied. And so I went and interviewed them. And in all cases, I decided they don't get it. They don't understand it. And I decided instead at that time that it was too early to really get into it, to get into aging. And so I went into biotech instead and worked on cancer research, heart disease research, and inflammation research, and a few other things. I mean, as a result, I became National Inventor of the Year in the United States in 1997 for my cancer research. I made a lot of big, I'm responsible for a lot of the big biotech breakthroughs, including as far back as human growth hormone and tissue plasminogen activators, so heart disease and cancer drugs and stuff. But my heart was always in aging. And so I was always looking for a way to do it. And my father, who was a television producer, even decided he was going to do a documentary on aging to try to get some interest, and so that maybe I could do something. But it never happened. But then I attended a conference that in 1993, I think it was, that where I heard a speaker speak on something where the 2s and 2s did add up. And I'll be coming back to those during this series. And I went instantly and started working with the guy, made a lot of success. Then 4 years later, started my own company and have been continuing all that research ever since.
Fantastic. So yeah.
And I'm also an ultra marathon runner like you are.
Oh yeah, an ultra marathon runner like me. Yeah, another crazy person who's done extreme stuff. But that's you. You're extreme, and you don't let limits stop you. You don't let people, when they tell you can't do something, be the final say. And that's really a strong trait of your personality. And you have to be a bit like that, don't you, if you're going to cure aging?
Somebody tells me something's impossible, it becomes my main obsession to prove them wrong.
Absolutely. And part of your PhD, Dr. Bill, was in evolutionary biology as well. Can you give me a bit of a reason why the heck do we age, and what is aging?
Well, it's the why, and the why is going to be part of the next series, but let me just scratch the surface for a second. Let me start with first saying
what is it? Yeah.
What is it?
What is it?
That'll be easier to go into the why. And aging is pretty much just the decline and failure of multiple body parts. Or the body parts are tissues and organs. So it's a failure and decline and failure of multiple tissues and organs. We all are passing away because some organs, more than one usually, are just failing. And the question of why they fail is a question for the next series, but it pretty much has the fact that all tissues and organs do have what are called they're the cells that are doing all the activity, okay, of the organ. And because of their activity, there's a lot of wear and tear. And as a result of wear and tear, those cells get destroyed. They're removed from the organ or tissue, but that's okay because other cells inside the organ and tissue can divide to replace those cells. Okay, so given just that, we should be immortal. We should always be just being able to replace, repair our organs and stuff with cell division. But the problem is that our cells can't divide indefinitely, and I will be talking about that. And that's when they start running out of the ability to be able to divide to replace the damaged cells. That's when the organs start declining and failing. And that'll be, I'll spend a lot more time on that later. But that's why, and it's the decline of the multiple tissues and organs that gives us all the appearances, both the way we feel, the way we behave, and the way we look about aging.
Yep. Okay. So, and when we look at it from an evolutionary perspective, why do you think that all the successful species that have dominated the planet, or most of them, have this aging process that's linked to telomeres, which, you're a telomere scientist, we're going to get into what telomeres are, but what's the connection to evolution?
It's actually not true. They're not all associated with telomeres. Telomeres, I'll talk about this later too, but telomeres are really playing a role in aging in only dogs, cats, horses, sheep, pig, deer, some non-human primates, and humans. Okay, other animals, telomeres don't have anything to do with aging, but there's still other mechanisms that are causing the cells not to be able to divide to replace the damaged cells or the lost cells. So, but so the why is again something that will be in the later series, but it turns out to be that people have a hard time accepting. There turns out to be an evolutionary advantage to the species to eliminate the longer lived. Okay, so by longer lived, I mean if you've raised your young, you are then considered a longer lived. You offer nothing to the survival of the species after you've raised your young. The shorter lived are the ones that haven't finished raising their young, and it's very important that they survive in order for the species to survive. Okay, so once we've lost the need to be alive to help our species, then just accumulation of things. I'm getting too much into the next parts of the series, but there's just an evolutionary advantage to living longer than it takes to raise our young. That's the bottom line.
So we needed to mix up the gene pool, and if we kept the older people around and they were as strong and as vital as always, and were reproducing, we would have had problems with our gene pool way back in the day.
Well, let me say another way. Before sexual reproduction, there was no aging. Okay.
Wow. Okay.
Aging only became an advantage to the species after sexual reproduction, and because of sexual reproduction, for the first time, we started shuffling our genes.
Yeah.
And which created more diversity within our species, which gave us a higher chance of surviving. I'm getting way too. We're gonna not.
Well, we're gonna have to.
But that's pretty much the what aging is really simple. Getting back, it's just as I said, the decline and failure of multiple body parts or multiple tissues and organs, and that's what we have to solve. And there's a lot of different ways of solving it. I'm not going to say there's only one. I'm going to say that in the case of humans and the other animals I mentioned, it'll never be solved unless we also, in addition to anything else we have to do, we have to also solve the telomere shortening problem, because that's the one and only absolute thing there is about aging that there's nothing we can do about it at the moment. Okay, and it puts an absolute limit on our lifespan. Okay, which is 125 years, and none of us live that long because of poor lifestyle choices and poor genetics. But we have to solve the telomere shortening problem in addition to maybe some other things. And I don't know, maybe it might be the only thing we have to solve, but I'm not going to count on that. I'm glad there's other people like me, like Aubrey de Grey and Greg Fay and Mike West and other people that are working on trying to cure the aging process through other means that I think are very valid. But we'll get onto that. But I don't think anybody's ever going to really succeed unless they really first understand what aging is. And that's why I wanted to focus on that in this first series.
Yeah. Okay. So what? Yeah. What is aging then? And let's talk into the depths of why should aging be considered a disease, and why is it not considered a disease at the moment?
Well, it's not considered a disease because all of us have it. Okay. And I always say, just because all of us have it doesn't make it not a disease. Okay. But but at the same time, I don't really know if it's a disease. Okay. I just know that when somebody cures it, everybody will consider it a disease. Okay. But until we cure it, it's still a question, is it a disease or not? But I think there's a chance it is. Okay. Now, something else that contributes to that chance that it's a disease is there are life forms on this planet that have no detectable aging. Okay. Now, I'm not talking about trees or jellyfish or things like that, because those are so different from humans. I mean, a tree can be alive if one little branch in the very highest roots is still putting off leaves. That tree is considered alive, even though the rest of the tree is dead. Well, if my little finger was alive and the rest of me was dead, I would not consider myself alive. So humans are a little bit different than trees. Okay, now same with jellyfish. Jellyfish seem to be more of a colony of cells. Each cell is an individual life form. You can take a jellyfish, cut it in half, and each half will grow into a jellyfish. You can take a jellyfish and you can break it into all its individual cells, and those will all grow into jellyfish.
Wow.
Okay, so they're really kind of a colony. And humans, you can't do that with humans.
No.
You can't take a piece of your skin. Well, you can. Science has figured out how to do that now, but you can't. It won't happen just naturally.
Yeah, we don't grow a new arm if we chop it off or something like that, do we?
Yeah. And yeah, that's the other thing. People are now figuring out. I know people that have made some really good strides in figuring out how to grow your arm back, if that happens. But yeah, so we don't know if it's a disease. The fact that a lot of animals don't have, and the examples of these animals are lobsters. Lobsters have no detectable aging process. Humpback whales, clams, some fish, some birds, tortoises, they have no detectable aging. Now, unfortunately, most of these animals have no way, they don't have something equivalent to like rings in a tree. When you cut a tree down, you can count the number of rings and know how old it is. So there's no way to look at a lobster and say how old it is. But that's not true for clams.
Yeah, they got
clams do have stripes on their shells, and they get a new stripe every year. And so when people started realizing this and they started counting the number of stripes, they were finding clams that were over 500 years old.
Wow.
One most notably made a lot of headline news, at least in my field. And it was a female clam that they named Ming. Unfortunately, the clam was already dead because they harvested it for food. And it was only after they harvested it and it was dead that they counted the number of stripes. So they killed a 500-year-old clam.
Oh, no.
But they call it Ming because it was born during the Ming Dynasty.
Wow.
And but sharks are another animal. Sharks, you can tell the age of a shark by certain proteins in its eyeballs. They've been able to show that some sharks are over 200 years old. But most of the animals, nobody ever thought, well, how old can an animal live before the time of Darwin, let's say. So, but after Darwin, people started taking animals, putting them in cages and aquariums from when they were born. And then started watching to see how long they would live. And lo and behold, some of these animals just never showed any signs of aging. They're 200 years old. Like even Charles Darwin had a pet tortoise named Harriet. And she only died recently at like 180 plus years old.
Wow.
But she died from a virus or something like that, some disease, had nothing to do with aging. And there's another really famous tortoise that you see pictures of all the time. That's approaching 200 years old right now. And the only way they know it's 200 years old is because somebody was there when it was born. And it's been keeping it ever since. It was like born in a zoo or something like that. But then when these animals do die, they usually don't die from aging. They die from something else.
Yep.
So, yeah. So I think there's a high probability that aging is a disease, and I look forward to proving that by curing it. Then everybody will say it's a disease.
And that's what this series is going to be all about is, yeah, how do we cure aging? How far are we along in the science? What can we realistically expect to happen in the next 10, 20, 50 years? And have we got a chance of living a heck of a lot longer? Those are the questions that are floating around in our heads at the moment. You know, you're 72, I believe, and I'm 55. And we're like, you know, come on, Bill, hurry up. Sort
well,
of.
it sounded like you said I was 22, but I think you meant you said 72. Just maybe the accent.
Yeah. But you act like a 22-year-old. You're still running around or the planet doing crazy stuff. So maybe you've cracked the code.
Yeah. It's like, I want to say, I'm not afraid of dying. Okay, it's not a question of being afraid of dying. I just think of all the great things that are going to happen in the future, and I think it's a big adventure. You know, I don't want to miss out on all the great things. You know, oftentimes when I speak, I will start off by, because there was one time I spoke in India, and the audience was actually quite critical of me for being afraid of dying. And I was really surprised by that. And I said, I'm not afraid of dying. I mean, a lot of the things I do a lot of dangerous things, including ultramarathon running, which can be very dangerous. And if I was afraid of dying, I sure wouldn't do those. But, I've jumped out of airplanes. I've flown gliders and hang gliders. And I've done a lot of really crazy stuff, dived off big cliffs. It's like, I wouldn't do those kind of things if I was afraid of dying. What I'm afraid of is missing out on all the great things that are going to happen. And I will often show a picture of First Contact, was a TV series, one of the Star Trek movies. And they show the guys shaking hands, humans shaking hands with a Vulcan for the first time. And I paid a lot of money to have some artist create the photo with me in it, doing a three-way handshake. And so when I tell people I'm not afraid of dying, I'm afraid of missing out. When we make first contact, I want to be there, and all of a sudden I appear right in the photo, and that's the whole point. The big one of the biggest things about defining what aging is all the misunderstandings about the markers for aging. How do you measure aging? And there's a lot of people believing that their aging has been reversed because markers have shown that they have become supposedly biologically younger.
Yeah.
They don't look, feel, or behave, or they may look like they don't look and behave and feel younger. They may feel younger, but they don't in some ways, but it's sometimes it's just a improvement of health. Okay, there was some organ was failing, and they made that organ feel better, or that tissue was starting to fail, and that tissue became better. That didn't mean they reversed their aging. So a lot of people have come to believe that you can measure aging by measuring the length of telomeres, or measuring the amount of DNA methylations that are on your DNA, or measuring certain proteins like glycosylation of certain proteins. Those are really, really good markers. But are they measuring aging, or are they measuring some aspect of your health or something? And are they, so I always say, what I do is, when I say none of those, we don't really know if they're really measuring aging, because how would they know? How would they know that they're measuring aging? They can correlate with aging, but that necessarily isn't a measurement of aging. And I always say that the only way we will ever know if we have good measurement of aging is to be able to reverse aging and see if that measurement correlates or corresponds with the reversal of aging. But then there's a kind of fallacy, not a fallacy, kind of like a logical problem. There's, how do you know if you've reversed aging if you can't measure aging?
Yeah.
And so I always show a picture at this point in a talk. I'll show a picture of Betty White, movie star Betty White, when she was 25 and 95. And I just say to the audience, is there anybody here who doesn't know which photo of Betty White was taken first? And it gets a good laugh, but you know, was it the wrinkles? How does everybody know which photo was taken? Was it the wrinkles? And I'll say, you know, imagine getting rid of those wrinkles. You'd still know that photo, the 95 was taken later than the photo of the 25. I mean, there's something about the appearance of a person that can make any of us be able to guess within like plus or minus 5 years of how old that person is.
Yep.
None of us can actually can say exactly what it is, but we know we can do it. So I always call it, we can't quantitate aging, but we can qualitate aging. And I can also say by taking several different people, and then we can say, you know, put a bunch of people in a room and say, okay, have everybody line up, and then you put them in order of age. You'll be pretty good at that. So you're kind of quantitating at the same time as you're qualitating. So I always call that being qualitatively quantitative. Okay. Yeah. It's like I give people what is called a QQ score. Okay. So they have a QQ score of being between this person and that person, you know, and that kind of stuff. But we won't really know anything about what is causing aging or how to measure it until we actually reverse aging. And so that's yet to come. And you know, that's not the main reason why I want to cure aging, but it's going to be one of the icing on the cake, is we're going to have better skills at measuring aging when we do that.
Yeah. And where do you think we stand with the science at the moment? You know, well, if we talk about your area of expertise, which is in the telomere side of things, you know, where are we currently? And what are telomeres?
I don't want to call my area, well, my area of expertise, let's say my specialty is telomere biology, but I study all aspects of aging. Okay, I believe they're all important, and so I stay on top of them. I'm a big follower of Aubrey de Grey and Mike West and Greg Fay. Well, Mike West believes a lot in telomere biology too, but others believe other things, and I'm following all of them, and I kind of, when I have the opportunity, do research in all of them too. And I'm scientific advisors for companies that work on aging that have nothing to do with telomeres, and they believe they can reverse aging. And who am I to say they're not? You know, we don't know. I'm a scientist. I don't say I believe this to be true or that to be true. I just go with the data. Okay, and right now, nothing, not even telomere biology has been shown unambiguously to be able to reverse aging, even though we have it does show the best results, I think, so far, because it's the only thing, lengthening telomeres is the only thing that's been able to reverse aging in human cells in a petri dish by every method of measurement imaginable. It's been able to reverse aging in human skin grown on the back of a mouse by every method of measurement imaginable or available. And also, like when I mentioned all the names of the animals that age by telomere shortening, you might have noticed I didn't mention mice or any rodents.
Yeah.
They don't. And but people have created mice that do age by telomere shortening, especially Dr. Rhonda Pennell at Harvard when he was there. And he was able to show reversal of aging in every way he could measure when he lengthened the telomeres in these engineered mice. And there was people can do a Google search and find a really good interview by news reporter Diane Sawyer, where she interviewed Dr. Rhonda Pennell and went over the data, and she looked at the mice and things like that. And he referred to it as a remarkable reversal of the aging process. Now, nobody else has done that with any other theory of aging. They've been able to say they've reversed aging by showing that DNA methylation patterns changed, or glycosylation patterns on proteins changed. But and then there's at least 50 biomarkers of aging. I don't want to just pick out the two, but the none of them have actually been seen to actually cause every marker to show a reversal of aging, and then also the look, feel, and behave, like was seen with the mice.
So, but Dr. DePino did actually show. So his experiment was in mice that had, because mice, as you say, don't have shortened telomeres. That's not how they age. They age by oxidative stress and inflammation, I believe.
Mitochondrial dysfunction.
And mitochondrial dysfunction, yeah. But he made a specific type of mouse that did age through telomeres and showed that could reverse that.
Yep, it was a really good experiment. It's just very expensive, very difficult to do, and nobody's actually repeated the experiment.
Okay.
Partially, I think, because nobody questioned it. Dr. Ron DePino he's like one of the top scientists in the world. He actually became the head of MD Anderson, which is one of the most famous hospitals in the United States after that research.
Wow.
I believe he was trying to show that telomerase causes cancer. I don't think he was trying to show that they were going to get a reversal of aging. I think he was trying to show that he was going to give as soon as he induced telomerase to turn on and lengthen telomeres, he was going to see all of them get cancer. But they didn't. They got a lower incidence of cancer. And he was a cancer researcher, and I think that kind of blew his whole career. So that's why he quit doing research and went to work as the head of MD Anderson, but still focusing on curing cancer.
So it could, we just backtrack a little bit and just define what a telomere is. Because we sort of jumped into telomere shortening and it's being a problem. Maybe explain the Leonard-Hayflick and the Hayflick limit and telomeres and what they are exactly.
Okay, well, let's start with Leonard Hayflick. So before Leonard Hayflick, people used to think that you could take a human cell from a person, put it in a petri dish, and start to grow it, let it divide and divide and divide, and it would divide indefinitely. Okay, and just like you can do with bacteria and some forms of fungi and yeast and things like that, you can keep a culture going on forever and ever and ever. But Leonard Hayflick showed in the 1960s that wasn't true. He would take some fibroblast cells, which are skin in the dermis part of your skin, cells in the dermis part of your skin. Take some of those cells and start growing them in a petri dish. And then, constantly measuring how many cells there are, taking a small fraction, reseeding a new petri dish, letting those grow to what's called where they fill the petri dish, and then take a few of those cells, count them, put them in another petri dish. And how many times he would do this over and over and over again? We found out that when he took this original cells from like a newborn baby, they would divide 50 times. If he took DNA from or cells from like a 50-year-old, they would divide 25 times, and before they would stop. And if they took cells from a 95-year-old or 100-year-old, they'd divide less than 10 times before they would stop. And so suddenly he realized that there was a limit to the number of times a human cell can divide. And that became known as the Hayflick limit. Now, unfortunately, everybody thought he was an idiot. That couldn't possibly be true. He just didn't know how to do science. But I'll tell you, he's probably one of the best scientists there are, because believe it, opening up a petri dish and taking cells from one petri dish and putting them into another over and over and over again is a very, very difficult thing to do. You have to be a very good scientist because of the risk of contamination. As soon as something else in the air or something contaminates that petri dish, experiments over. Okay, and so to be skilled enough in sterile technique to be able to do that over and over again without ever getting contamination shows that he's a truly great scientist. And most people that tried to reproduce it couldn't do it, not because they showed that there was no limit, but because they couldn't reach that limit because of contamination problems. So Leonard Hayflick did that, but no, Leonard Hayflick had no idea why those cells had a limit. How could a cell know how many times it has divided? Or how could a cell know how many more times it can divide? And that was a big question that everybody was saying, that's impossible. How could a cell know? But back in the 1970s, when I was still in high school and going to college, this is when I first learned about the Hayflick limit. So I was about 10 years later. And my first thought was, no, that's not wrong. That's probably right. Because I always thought that there had to be some counting mechanism for aging that would make it, because the other thing is, again, I'm really strong in statistics and statistical theories and logic and things like that. And so with all that thinking and all my anti-aging clubs I used to participate in, the question always came up with, well, if aging was just a random event of nature and environment, why is the curve so narrow? Why do we see such a, it's not a big normal curve of like how old we are when we die of old age. It's actually very offset to us, like peaking at about the late 70s, okay, and then a very narrow range. So there had to be something else going on besides just randomness. And I used to always say there had to be something like ride tickets at an amusement park that our cells have inside of them.
That's a great analogy.
Every time a cell divides, a ticket's taken off. And I knew that cells had to divide, and because I knew that, and I've known for a long time that the average lifespan of any cell, the word I was looking for before was frontline cells. So tissues and organs have some cells that are part of those things that are called frontline cells, because they're like on the front of the battle. They're where all the actions occur. We know that the average lifespan of those cells are 1 year. Okay, and so something has to replace them, and they can only be replaced by cell division. And so I would always imagine there was some ride tickets at an amusement park that every time a cell divided, it ripped out the ticket.
Yep.
And then one day I heard somebody talking about telomeres. Telomeres are at the very tips of your chromosomes. They're like the caps to your shoelaces. Okay, so your chromosomes are really like linear things like shoelaces are, except when you see them in a microscope, they're all coiled up like a slinky. So take your shoelace and roll it up like a slinky around your finger. Okay, and that's what a chromosome is. But at the very tips of the chromosomes and the shoelaces are the caps. Okay, and we know in the shoelaces, when the caps get short, your shoelace falls apart. Well, turns out the caps on our chromosomes are called telomeres. And when they get short, our DNA falls apart. Our cells die. Our cells lose the ability to function correctly. And so it's actually, in terms of markers of aging and stuff like that, telomere shortening is the only really marker that we totally understand its mechanism. Okay, we know exactly why telomeres shorten. It's actually, they don't shorten. They don't degrade, they don't wear away, they don't do anything. What happens is every time a cell divides and the DNA, everything inside that cell needs to be duplicated, so that the two daughter cells have the same everything as the parent cell did. One of those things that has to be duplicated is the chromosomes or the DNA. And so it turns out that when a human cell duplicates a DNA, it goes along, like, let's say this shoelace, let's say it starts in the middle and goes both directions, duplicating this shoelace, or in the case DNA. But when it gets to the very ends, it can't duplicate the very ends.
Very end, yeah.
The new shoelace or new DNA is made shorter.
That much
Okay.
shorter. Yeah.
And that's the shortness is the ride ticket. Okay, it can't duplicate the final ride ticket. And we know exactly why it can't. And we know that I use an analogy of being a bricklayer
Yeah.
and standing
Good.
on top of a wall, backing up, laying the next row of bricks. Okay, and then when you get to the end of the wall, you fall off without being able to, before you can
Place
replace
that last brick.
with that last brick. So the top row of bricks is shorter than the previous row. That's almost amazingly 100% accurate as to what's
Yeah.
going on in the humans.
There was a little video somewhere that I saw that you'd explained that as the bricklayer can't stand on the last step, and so it gets shorter. So these telomeres get shorter. And when we're conceived, we have 15,000 base pairs, which is the measurement used for telomeres.
Yeah.
And then what happens, so from the moment of conception, we've got these hugely long telomeres, 15,000 base pairs. What happens then?
Well, every time the cell divides, so the cell becomes, it starts off as one cell, then it becomes 2 cells and 4 cells and 8 cells. Every time that divides, you lose a ride ticket. Okay, ride ticket is about 50 to 100 bases in length. Okay, so when you're first born, so when you're first conceived, your telomeres are 15,000 bases approximately. When you're born, you've already gone through so many cell divisions that you're already down to 10,000 bases. You've lost 5,000 bases telomere already by the time you're born.
Wow.
And it doesn't stop there because you still have a lot of growing to do. You've got wounds to heal. You've got infections to fight. All these things cause cell division. And so your cells are dividing, and so you're born at 5,000 or 10,000 bases. And when the telomeres get down to 5,000 bases, the cells lose the ability to function. They don't get it to 0. It's at 5,000 bases where suddenly all havoc is wreaked in the cell. The cell loses control of all its epigenetics, its gene expression, its mutation rates skyrocket, incidences of cancer skyrocket because of mutations. And the mutations are so major that you can see them in the microscope, a regular light microscope, whereas you can't see a base in a microscope, but you can see the chromosomes rearranged in the microscope. It's a thing called chromotherpis, and I'm not sure I'm pronouncing that correctly, but it's like chromotherpis. And so that's what happens. Now, if we could keep the telomeres long, none of that would happen. But
so when a cell, so like this is a cell and it's got its DNA and it's got its telomeres on the end, and they get shorter and they're getting down to 5,000. But this is happening in every single cell. But it's not happening at the same rate within. So even within, say, a liver, and you've got I don't know how many billion cells in your liver. Are all of the telomeres shortening at the same rate, so the liver then gives out all at once, or is it being replaced by stem cells? And do stem cells then have longer telomeres again, or no?
well, I'm going to divide the cells into 3. There's progenitor cell, there's stem cells, progenitor cells, and frontline cells. Okay, so the best way to think of stem cells and progenitor cells are like the reserve cells. So if we go back to the military kind of analogy, the front line is who's doing the battle, but behind them are the reserve soldiers that are going to be going to the front line when somebody in the front line is killed or something like that.
Yep.
So the same thing is happening in all our tissues and organs. There's the reserve cells, which are the stem cells and progenitor cells. So progenitor cells are made from stem cells. Progenitor cells are the precursor to the final cells.
They're more differentiated than the stem cell. Yeah.
And so I guess the frontline cells sometimes are called terminally differentiated.
Yeah.
But they don't always have to. Okay, so so during this whole process, the frontline cells, of course, they're the ones dying off first. So their telomeres are going to be a different length than the progenitor cells and the stem cells. But ultimately, when the serious problems happen is when the stem cells telomeres get so short, they cannot create the progenitor cells anymore.
Right.
And then when the progenitor cells telomeres get short, they can't replace the frontline cells anymore. That's happening. But there's different lengths of telomeres in every organ. That's why measuring telomere length is not a really good measure of aging, because it's so difficult to do. Okay, it's like when you look at measured telomeres, you actually get a whole range of telomere lengths, and people just go with the average typically.
Yeah.
And they're very difficult to measure. Now, that said, some organs will have entirely different length telomeres than other organs. Okay, so let's take a person who's got muscular dystrophy. Okay, well, they have a mutation in a gene called dystrophin. And that dystrophin mutation, and I don't want to call it a mutation, let's call it a gene variant, because we don't know if it was a mutation. Maybe they just inherited it from parents and something like that. But the dystrophin gene variant causes the muscle cells to die faster than once a year. Okay, so the progenitor cells and stem cells have to divide more often to replace those lost muscle cells, the frontline muscle cells. And as a result, you'll have faster telomere shortening in your muscles than you will in your other organs. And so the first organ, or first tissue, this would be tissue anyway, to fail is going to be the muscles in a patient with muscular dystrophy. And since some muscles are essential for survival, you're going to die from the failed organs. Okay, you didn't die of old age, because in this case it was only one organ. That's why I always say aging is when multiple organs fail.
But it's the same process of telomere shortening being caused by this gene variant in this case.
The gene variant did not cause the telomere shortening. The gene variant caused the need for the cells to divide more often to replace the killed cells. And as a result, the telomeres got shorter faster because of the cell division.
Gotcha.
Now, there's other kinds of cells like neurons that never divide. Okay, of course, they have a history of cell division ever since the single cell embryo. They've been dividing, but they hadn't quite differentiated into neurons. But when they do become neurons, the telomeres in those neurons are already shorter than they were. They're already shorter than 15,000 bases. And, you know, I don't know if anybody's actually done a study on looking at that, but they don't divide anymore. Okay, the only way telomeres are going to shorten in the neuron cells is from oxidative stress, or may actually only oxidative stress or some type of DNA damage, things that cause damage to the DNA. Now we know that that's, in long run, a very minor cause of telomere shortening relative to cell division. Cell division is the major cause of telomere shortening. But in the case of neurons, people who study neurons, they find that neurons are the most healthiest cells in the world. Neurons can far outlive a human. You know, I said frontline cells last about once a year. Neurons, they last hundreds. They could last hundreds of years and be perfectly healthy. And that's because they have all these caregiver cells and bodyguard cells that take care of them, like oligodendrocytes and Schwann cells are caregiver cells, because they are continually wrapping the neurons in something called myelin.
Yeah.
Protects these neurons, takes care of them. Then we also have other cells like astrocytes, which are kind of like immune cells. I call those the bodyguard cells. They're protecting the neurons. Well, all these cells that I mentioned besides the neurons, so the oligodendrocytes, the Schwann cells, the astrocytes, they all have to divide to do their job. They do have wear and tear. They do get replaced once a year. They do have telomere shortening. And so when their telomeres get really short, that's when a lot of dementia type problems start occurring.
Wow, so it's not the neuron per se. Generally, it's like either the myelin sheath or the astrocytes or
Yeah, well, the neurons start experiencing wear and tear because they don't have
not looked after.
caregiver cells and the bodyguard cells to protect them. And so but it's not the only thing. There's mutations, for instance, that can make neurons have a low survival anyway. So it's not just telomere shortening, but neurons typically have longer than normal telomeres, and then really never experience telomere shortening because they're terminally differentiated, never divide. Of course, you can always make new neurons, but not so easily. But it's a whole different animal in terms of inside of the human body. Neurons
slower in the brain to deteriorate. Yeah.
It's not like muscle cells that can be explained by muscular dystrophy being explained by telomere shortening. Neurons, their decline is mostly due to the decline of other cells, the bodyguard cells in the front line, and the bodyguard caregiver cells and the bodyguard cells.
Yeah, yeah, and things like immune activation in the brain, the glia and things can get grumpy and start causing havoc and things like that as well.
They're all glia. Some are microglia, some are macroglia, and some play different roles than others. But yeah.
So so different organs will, because I've heard you say at one point, we've got all these fuses burning, and it's just which one is the shortest fuse is going to get you.
Yes.
So is that, like depending on your genetics, depending on the variants you have, the environment that you're in, that contributes. So apart from the Hayflick limit, which says like we can only divide so many times and then you're going to die, most of us don't get to that Hayflick limit, if you like, because we die earlier of another dynamite stick blowing up before we get there. Is that right?
Well, it's questionable. I mean, it might be that, oxidative stress and inflammation does cause wear and tear to the cells. But you would think that wouldn't matter as long as there were other cells that could replace them. So the end result might always be that the failure is because of telomere shortening. The exceptions, the main exception is cancer. Okay.
Yeah. Yeah.
Because in the case of cancer now, the mutation doesn't really cause the cell to divide. Cancer, the mutation causes the cell to actually grow. Okay. And so lengthening the telomeres isn't going to help you cure cancer. Okay. Except lengthening telomeres will help the immune system fight the cancer. And our immune system is one of our best defenses against cancer.
Absolutely. Yeah. Yeah.
But it turns out, if the cancer is big enough to detect, okay, to be able to visualize it, like see, then that has already overcome its telomere shortening problem, because the cancer cell will actually succumb to old age because of the division causing accelerated telomere shortening. They'll succumb early on before they're actually big enough to detect. If the cancer starts when you're like over 35, okay. If you're young, if you get a cancer when you're 10 years old, that cancer will get so big before telomere shortening can shut it down. It'll get so big that it'll kill you beforehand. So, so that's a different thing. But cancers can when you get a mutation that causes cancer, they experience incredibly short telomeres after they've divided it a number of times, and because the incredibly short telomeres cause mutation rates to skyrocket. Mutations will cause the telomerase chain to turn on, or another method called ALT, which stands for alternative lengthening of telomeres. It's the ALT pathway. an alternative pathway, another way of looking at it, that lengthens telomeres without telomeres. Those two type of things can result from all the mutations caused by short telomeres. And suddenly the telomerase is turned on, and then the cancer just becomes immortal and takes over the body and kills
all
you.
its resources, because this is and you've been a cancer researcher, done lots of cancer drugs and so on. And when you discovered telomerase, which is an enzyme that actually lengthens telomeres, you actually worked for a while on a cancer drug that would, sorry.
I did a lot of cancer research before I got involved with telomerase. I was actually National Inventor of the Year in the United States for my cancer research in 1997. So I do understand cancer a lot. I do want to qualify one thing. I did not discover telomerase. I discovered human telomerase.
Right.
Okay. And I discovered what it is. It was known beforehand that telomerase existed, but nobody knew what it was. They knew that somehow our reproductive cells, when they divided, the telomeres didn't get short.
Yeah.
So something was re-lengthening the telomeres every time cell division would cause them to get short, they get re-lengthened. And it's like a tug of war. And that turned out to be the enzyme telomerase, which in humans I discovered. It was already discovered in other animals like tetrahymena, which is a pond scum organism that doesn't have any aging, but they still divide, and so they constantly have to be re-lengthening the telomeres. So that was discovered by Elizabeth Blackburn and Carol Greider and Jack Stosack in the 1980s. And after all the work that we and everybody else did on human telomerase to show how important it was, the Nobel Prize Committee decided somebody needs to win a Nobel Prize for this. And the way Nobel Prizes are, whoever originated the field gets it. So the Nobel Prize went to, I think it was 2009, went to Elizabeth Blackburn, Carol Greider, and Jack Stosack for their pioneering work on telomeres and the importance of telomeres and telomerase.
Yeah, and then the false, because I've listened to your lecture in Tokyo where you talked about because then there's been this, well, rumor, I want to say, that telomerase, then if cancer cells are showing that they have a lot of telomerase, they're actually immortal because of this, then it would be bad to put in telomerase because that would cause cancer. And that's a bit of a logic problem, really, isn't it?
Yeah, it is. And I've talked in Tokyo many times, but you're referring to the Tokyo 2017 presentation I had on my website. Yeah. And I explained very carefully why there was a logic problem there. Well, two things. If you're an expert in logic, you know if A causes B and A causes C, it doesn't mean B causes C.
Yeah.
Okay. And that's the major logic problem that's occurring with a lot of the stuff that's going on. With a lot of people who think that telomeres and long telomeres can cause cancer. But what is known is that the cancers actually cause the production of telomeres. A cell has to become a cancer cell first. And some people like to call it just a tumor. Okay, so a tumor is essentially a cell that's not going to be a cancer, but the cell loses growth control. Suddenly, like I always use Pinocchio's nose as an example. Our noses grew to a certain length and stopped. Okay, they stopped because of growth control. Okay, something tells the cells in our nose to stop dividing. Mm-hmm. And when you lose that growth control, your nose grows like Pinocchio. Mm-hmm. Okay, so the way that you lose that control is from a mutation. The mutation either interferes with what's called a tumor suppressor gene or an oncogene that causes cancer. And so first of all, you have to have a mutation that causes cancer, or causes uncontrolled cell division. And a lot of times those get destroyed because the telomeres end up killing all the cells. And so this cancer never gets big enough to detect if it starts after age of like 35. But the problem is that in addition to the telomeres getting shorter and the cells losing the ability to function, the mutation rates are skyrocketing. And so the mutations cause a mutation that turns on the telomerase gene or turns on the alt pathway. And then that cancer becomes immortal. So the telomerase didn't cause the cancer. The cancer caused the telomerase.
Gotcha. It's a backwards thing. So it's chicken
Yeah.
and egg scenario. So at the
Yeah.
beginning, though, the
Part before the
telomere,
horse.
yeah, the telomeres being shorter at the beginning, like when they get short, that causes more mutations, and that
Yes.
causes cancer. So short telomeres cause cancer,
Yes.
and then the telomerase turns on once it gets to a certain size, and it mutates more, and the telomerase then comes into the party. But it's not when you go and use telomerase inducing supplements, drugs, which you and your company are working on, you're not causing cancer.
We're decreasing the risk of cancer.
You're decreasing the risk of cancer.
By inducing telomerase before a cell becomes cancer, before it loses growth control, makes it very unlikely that it'll have that mutation that causes it to lose growth control.
If you already have cancer, is it?
If you already have cancer, those cancers have already solved the telomere length.
So you're not going to impact that by taking a telomerase inducing agent of some sort.
You can't make something that's immortal more immortal.
More immortal.
Now there's going to be those rare exceptions where the cell has lost growth control, but it has not mutated to turn on telomerase. And in that case, the telomerase might allow those cells to get bigger and bigger. But it's also, telomerase is also causing the immune cells telomeres to get longer, which increases the immune cells' ability to fight the cancer. And as I said, our immune systems are best defense against cancer. Our immune systems are actually pretty good. Cancers don't overwhelm the immune system until they get big. Okay, so really, really small cancer is very unlikely. I don't even want to call it cancer yet, because terminology, even cancer doctors use different terminologies to define what the word cancer means. Let's say a tumor that's not immortal, okay, it's going to be very unlikely that that's going to overwhelm the immune system. Okay, the overwhelming of the immune system occurs from all the kinds of cell divisions that are going to occur because of it's already turned on telomeres. Now they start developing mutating every kind of mechanism imaginable to even prevent the immune system from fighting. But none of that happens if you just keep the telomeres long to begin with. Okay.
Exactly. Yeah. So
it's rare because we know that some kids get cancer when their telomeres are still long. So it's still, there's always a chance that some kind of like photon from lights or some radiation or something like that is going to cause a mutation that gets cancer. So it's just far less likely to get cancer when you're young with long telomeres than you are when you're old with short telomeres.
And this is why we see a much bigger increase in cancer. And all aging diseases we see, as we age. I mean, they're aging diseases. The cardiovascular, the diabetes, the dementia, all of these things happen more and more as we get older and older. And this is a piece of that puzzle, that the telomeres are getting shorter and causing all sorts of havoc in this chaos in the chromosomes. And the reading of the DNA, because we are methylating our DNA and we have the epigenome. Can you explain what the epigenome is in relation to the DNA? So you got DNA, everyone sort of gets that double helix strand with the code that you got from mum and dad, but not a lot of people understand what the epigenome is and where is the epigenome and what is it and how does that play into all of this?
Well, epigenome is almost like a virtual, or it's not exactly a physical thing. Okay, so here's what happens. Okay, so along our DNA, we have our genes. Okay, and that's pretty constant. It's pretty much the same in every cell of our body. Okay, there's a few exceptions like our immune system genes. Our immune genes will change from one cell to another.
Wow.
But every gene is responsible, has a role, produces something, plays a role in the cell's health and function and everything like that.
Mhm.
And every gene has an on-off switch. Okay, and they're more like dimmer switches. Okay, they can be all the way off, they can be partially on, they can be completely on. And that depends on how much, like hair color, eye color. Those are controlled by genes. Okay, and so let's say black eyes. Black eyes are going to be really black if that gene for black eyes is really turned on strong. But if it's completely turned on, if it's completely turned off, the person's going to have blue eyes. Okay, so okay, so the epigenetics. Okay, so when a gene is turned on, it produces what is called an RNA. Okay, and that RNA then is responsible for producing the protein. If the protein is going to be the final product, sometimes the RNA is the final product. The protein then plays a role in the health. Sometimes the RNA actually plays a role in the health. But at any particular time, you can take a bunch of cells and you can look at how many different RNAs there are in the cells and be able to say which genes are turned on and which genes are turned off. So epigenetics is the study of which genes are turned on and which genes are turned on. Okay, so the epigenome is going to be the epigenome. So the regular DNA genome does not change or barely changes. The epigenome changes all the time.
All the time. Mm-hmm.
The environment changes it, genes are turned on and off, aging changes it. So the epigenome is going to be what RNAs are present and what abundance are they present. Now, telomeres actually play a role in that too. So telomeres actually play a role in turning those light switches on or off, or turning them to halfway on and off. And the shorter a telomere gets, the less likely it is able to reach over to those genes that are distal. Okay, and so if any of your listeners know what enhancer sequences are, enhancer sequences are components of DNA that can be like 10,000 or more bases away from a gene. The DNA folds over and brings this enhancer in contact with the light switch, which is called a promoter. Okay, comes in contact with its promoter and turns it on or off. Okay, telomeres can act just like telomeres have all the hallmarks of an enhancer sequence. They can fold over, they can come in contact with promoters and turn them on and off. And the shorter they get, the less far they can reach, or less number of genes they can reach at the same time.
Wow. So they are part of that turn the switches on, turn the switches off process, not just the RNA and the DNA methylation patterns per se.
But epigenetics, so people often call the epigenetic marker, they say DNA methylation is an epigenetic marker, but it's actually a mechanism that controls epigenetics. It's not actually epigenetics itself. Okay, so the DNA methylation, just like telomeres, can turn genes on and off. Okay, but that's all they do is turn genes on and off. The epigenetics is the measurement of how much the genes are turned on and off.
Gotcha.
And DNA methylation is not measuring that. It's just measuring the DNA methylation. It's just measuring how many methyl groups are on the DNA.
Uh-huh.
The only really good way to truly measure epigenetics is what's called total RNA sequencing. And there's companies that do that, like a company called CD Genomics. You can send in your cells to CD Genomics, and they will send you back a list of all the different RNAs produced in that cell and their relative abundances.
Wow.
And that's probably the most important health marker I can imagine.
Wow.
Is, and that's something that we do here too for our research at Sierra Sciences.
Mm-hmm. Mm-hmm.
We
Wow.
do a lot of our studies with, when we're changing telomere lengths, we're looking at the epigenetics by doing total RNA sequencing.
So at Sierra Sciences, you have spent, well, more than a decade, or I don't know how many, 20 something years, looking for telomerase inducing agents, and you've got thousands, 50,000, I believe, agents that you've found that help. And was it 900 in the nutraceutical side?
It's 37 in the nutraceutical side and about 900 in the pharmaceutical side.
900 in the pharmaceutical side. So these are things that are going to actually help lengthen telomeres, but you haven't found any that's going to have a massive impact. So you described it to me like you're sort of on a tug of war, and you can add telomerase back, but you're still adding back less than you're losing at this point in your research.
Lengthening and shortening is like a tug of war. You've got the shorteners pulling to the shorten, you've got the lengtheners pulling to lengthen. The only lengtheners are really telomerase. Okay, so the more telomerase you produce, the more lengtheners you have. But we have not found a way to produce enough telomerase to win that tug of war yet, except gene therapy. Gene therapy is how we did all the experiments I mentioned before, showing age reversal in human cells in a petri dish, human skin grown on the back of a mouse, and then engineered mice. That was all done by a process called gene therapy, which is enormously expensive and is used today in like companies like BioViva are using it for.
Liz Parrish's, yeah.
They're using it for lengthening telomeres, but it's extremely expensive, and it's pretty much still a research project. And
it's experimental at this point. Yeah.
Lots being learned from the work that BioViva is doing.
Yeah.
We gotta find better ways of delivering the telomerase gene to all the cells of the body, and or figure out a way of turning the gene on to higher levels, the gene that's already in our cells, turning that gene on to higher levels so it can win the tug of war.
Yep. And in our fertility cells, it's a sort of an even match, isn't it? That's why we can have babies who are genetically or telomere-wise young. And yes, even if you're an older parent, say you have a child at 45 or something, or 40, your baby's not 40 years old. Your baby has brand new, and that's how we've, and that's because of the telomeres on that baby's DNA are at full length again.
Now, that's pretty much true 99% of the cases, but there are cases where one of the parents might have a inactive or partially active telomerase. And as a result, the telomeres in the sperm or the eggs, depending on some other father, will be a little shorter. And as a result, the newly conceived embryo will actually have shorter telomeres. Wow. And so will be born essentially older. And then when the baby's born, it might look normal at first. He or she, I don't want to say it, but he or she will be look normal at first. But if they have enough telomere length to at least become born,
right.
Then they might show what's called accelerated aging, and the most famous disease is called progeria. Huntington-Gilford disease, where children die of old age before they're 20. Okay, and they have all the same symptoms as a regular normal person who dies of old age in their 70s or later.
Yeah, horrific. And that's just because that one of the parents had a telomerase problem, basically. It wasn't.
This is actually a point of big mistake that a lot of researchers make. People will look for that mutation in that child, but the child may not have inherited the telomerase variant. They just inherited the short telomeres. And so really the studies and there are a few labs that do it right. The real key things that when somebody has a accelerated aging disease caused by short telomeres, they need to look at the parents, not the patient, okay, to find out the cause of their disease.
Yeah, and there was a famous case, I think, in Scandinavia somewhere, isn't it, where they
Yeah.
could trace that back in generations, and it sort of skipped generations and then came
You,
down.
you have listened. You've listened to more of my videos than I realize. Yes, I talk about that particular case. I've become good friends with the key scientist there because it's the best example of what's they're called diseases of anticipation. So, the my great grandparent had a telomerase deficiency, and as a result, the telomeres of that person's kids were a little bit shorter, but they were still long enough so that they lived long enough to reproduce. But then their kids had shorter telomeres, and their kids had shorter telomeres.
Wow.
And the kids, after like the 5th or 6th generation, now had telomeres so short, they were now not getting old enough to reproduce. And they were suffering from, I think it was aplastic anemia. And there's also several other diseases, idiopathic pulmonary fibrosis and dyskeratosis congenita, that are all diseases of anticipation caused by being born with shorter telomeres.
Wow, wow. That's quite amazing. Well, Dr. Bill, I think we've come to the top of the hour for our session number 1 of curing aging, and we've done a deep dive into telomeres and what is aging. And in the next part 2 and part 3 and part 4, we're going to be diving deep into the why of aging and how we can act, what can we actually do, because at the end of the day, that's what we all want to know.
And I can guarantee to anybody who's listening, we only scratched the surface on those other things today. There's
Exactly.
a lot I'm looking forward to telling everybody about.
Yeah, so make sure you tune into part 2 shortly. Well, thank you, Dr. Bill, and we'll see you again soon.
Thank you.
Thanks so much for watching, and we hope you enjoyed this video. Please like, comment, hit the notification bell and subscribe, and of course share it with just one person who you think could really benefit from this content. Click here to watch last week's episode, and click here for my website, and click here to watch a video you may not have seen yet.